of our immune system, of the leukocytes, the white blood cells of our immune system, is to be able to differentiate and distinguish between the healthy cells of our body and the infected cells, as well as the different types of pathogens that can invade our body. Now, the question is, what exactly allows these leukocytes to not only communicate with one another, but also to distinguish between the healthy cells and our infected cells? Well, it's a complex of proteins found on the membrane of our cells that allows this process to take place. And the complex of proteins is known as the major histocompatibility complex, or simply MHC. Now, specifically in humans, because MHC is found in all animals, if we're talking about humans, then the major histocompatibility complex is also known as the human leukocyte antigens, and we'll see why it's given this name in just a moment. Now, there are three different types, three different classes of major histocompatibility complex. We have MHC class 1, MHC class 2, and MHC class 3. And in this lecture, we're only going to focus on MHC class 1 and MHC class 2. So let's begin with MHC class 1. And this is the diagram that describes this complex, this protein complex, found on the cell membrane of a healthy cell. So we essentially have 4 different polypeptide units that are connected as shown. And we have alpha 1, alpha 2, alpha 3, beta 2 subunits. And the alpha 3 is connected to the cell membrane of our cell. Now, what exactly is the purpose of the MHC class 1 complex? Well, basically, this is the major histocompatibility complex that actually allows the leukocytes, the white blood cells of our body, to distinguish the healthy cells of our body from the infected cells of our body, those cells that were infected by some type of pathogen, some type of virus, or some type of parasite. Now, most cells in our body that are nucleated, which contain a nucleus, have the MHC class 1 complex. So let's actually discuss how this complex works. Well, a healthy cell will bind one of its normal proteins, peptides, that it creates inside the cell onto this flat portion of our MHC class 1 complex. So if we take a look at the following diagram, we have a healthy cell of our body, we have our major histocompatibility complex class 1, and some type of protein that the cell produces inside our cell. It brings out and it binds onto this region right here. So when a leukocyte essentially approaches this healthy cell and it recognizes this peptide as a normal peptide, as a normal self-antigen, so we call this a self-antigen, which basically means that it's an antigen, it's a protein that is formed by this cell. So when the leukocyte recognizes a self-antigen, it basically leaves that cell alone. It knows that that is a healthy cell. Now, what happens if a cell is actually infected by some type of pathogen, for example, a virus? Let's suppose now we look at an infected cell. So the infected cell now contains viral components that have been created by that cell itself. So we have these different types of viral proteins. And what the cell does is it essentially takes a viral protein and it places on this complex here inside the cleft instead of this self-antigen. So now we have a pathogenic antigen, an antigen that comes from that pathogen, our virus. And now when the leukocyte swims by and notices this pathogenic antigen, it will bind to it and initiate a set of different types of immune responses that will ultimately destroy this infected cell. So we see that the major histocompatibility complex class 1 is actually responsible for allowing our leukocytes to distinguish our healthy cells from the infected cells of our body. Now, what about the second class? What about MHC class 2? Well, these are the complexes found on our cells, specifically actually on certain types of immune cells of our body, and these are used predominantly in communication between different types of leukocytes in our body. So these protein complexes are found only on specific immune cells, such as B lymphocytes, macrophages, our dendritic cells, and we'll discuss what these cells are in the next several lectures, as well as certain types of T lymphocytes. So basically, these look very similar in the sense that they both are composed of 4 subunits of polypeptides. But notice we have these homologous pairs. We have alpha 1 and alpha 2, and we have beta 1 and beta 2. And both of these are attached to our cell membrane as shown. And just like this one has a special cleft that can attach a certain type of protein, this one also has a special cleft as shown in the following diagram. So MHC class 1 are used to distinguish between healthy and infected cells, but MHC class 2 is actually used by the leukocytes to communicate between different types of leukocytes, and we'll see exactly what we mean by that in just a moment. So these protein complexes function in helping immune cells to communicate with one another. So let's suppose we have the following scenario. So suppose some type of macrophage as shown in diagram 1, so this is diagram 1. We have the macrophage, inside the macrophage we have the organelles, we have our nucleus, and we have these green structures which are the lysosomes. They contain digestive enzymes that can hydrolyze and break down the things that the macrophages actually engulf. So let's suppose we have some type of pathogen. Let's suppose a bacterial cell as shown in red. And the macrophage...
Okay, now he talks about these being simply macrophages. The same would hold true for if this were a dendritic cell or the helper T lymphocytes. Okay, so he's using it for macrophage, but those same markers are on several other types of cells as well.
Phagocytic cells?
Yeah, pretty much.
Which finds our bacterial cell and engulfs that bacterial cell. And so in diagram 2, we form this phagosome, this vacuole inside our macrophage, and these lysosomes fuse with this vacuole and they begin to break down that bacterial cell. Now, as they break down the bacterial cell, they essentially take some of those pathogenic proteins, our antigen, and they place the cell places our antigen onto our MHC class 2 complex, so the major histocompatibility complex class 2 as shown in the following diagram. So now that the macrophage actually contains this MHC class 2 complex along with this complementary antigen that came from our bacterial cell, it can basically release special types of chemicals known as interleukin-1. And what this chemical does is it goes on to recruit, it calls upon helper T cells. Remember, helper T cells are a special type of T lymphocyte. Now, once the helper T cell comes to this area, it basically has this receptor on the membrane that can recognize this antigen that is bound to the MHC class 2 complex. And once it binds, it begins to release its own chemical. Specifically, it releases interleukin-2. And what that chemical does is it moves on to B lymphocytes and T lymphocytes, and it forces these two cells to essentially differentiate into their specialized form. So B lymphocytes produce memory B cells as well as plasma cells, while these T lymphocytes produce the memory T cells as well as our cytotoxic T cells. So the cytotoxic T cells now contain the specific antibody on the membrane that can bind to this specific antigen. And so when the cytotoxic cell actually comes in close proximity to this type of bacteria that carries that same antigen, this antibody will be able to bind onto that bacteria, and once they are bound, the cytotoxic cell will begin to release these powerful proteins that will drill holes in those bacterial cells, killing off those bacterial cells. Now, we also produce the plasma cells, and what the plasma cells do, they essentially produce those specific antibodies that are complementary, that are specific to this antigen that was found on the MHC class II complex. And so we see that these MHC class II complexes are used by our leukocytes, by the white blood cells, to basically communicate between one leukocyte, let's say the macrophage, and another leukocyte, let's say the helper T cell, and then that can communicate with other leukocytes, for example, B lymphocytes and T lymphocytes, to produce even more specialized immune cells. So once again, to recap, the way that our immune system actually distinguishes between our healthy cells and infected cells is by using the major histocompatibility complex class I, so MHC class I. But the way our immune cells actually communicate with one another is by using a slightly different type of major histocompatibility complex known as MHC class II. So these are the ways by which our leukocytes are able to actually distinguish between healthy and infected cells, and the way that they can find these pathogenic cells inside our body.
Was that helpful?
I'm still kind of confused on class 2.
Okay, we're going to talk more about it, but that is the best explanation of class 1 I've ever found.
Yeah, no, class 1 made.
Class 1 makes perfect sense. Class 2, it's a little more complicated because it's an increased number of steps. But I thought he did a really good job going between what the two different ones did.
Is there another word for cytotoxic cells that you use, or?
Killer cell,
Natural killer?
Oh,
Natural killers?
now
No.
they-
no, they're not, no, because cytotoxic cells are basically activated T cells. They're a form of T cell,
So, okay.
okay? Yeah, they are a subset of T cells.
Okay.
So is class 1 essentially identification, class 2 is the communication and actual execution
Sort of.
in a nutshell?
Yeah, sort of. Class 1 is basically looking for infected cells. It's presenting from inside the cell. Class 2 is what we call the antigen presenter, where it takes the antigen and it basically says, here, this is the bad guy, this is what you need to make antibodies, or that the toxic cells are made against. And whether you get cytotoxic cells or whether you get antibodies, you're always going to get a little bit of both, but depending on what your pathogen is, I wish he hadn't used bacteria, I wish he'd used pathogen, because that's the only place that I think it would clarify it a little better, is that if you've got a fungus or you've got a parasite, that's going to probably be much more your cytotoxic cells. Those are the ones that are going to be much more functional in fighting those types of infections. Your bacterial antigens, the cytotoxic cells will help on those too, but you'll also get the antibodies that are made against them. Okay, and what happens, the antibodies get made, they coat the bacteria, and then your macrophages or your other phagocytic cells come through and eat them.
The complex,
Yes.
he showed it as, one little thing sticking out of the cell, but is it actually all around that?
Yeah, there's millions of them on the exterior of the cell. Yeah, it just drawed one because,
Well, yeah.
yeah. Yeah, no, that's actually a very good question, but yeah, they coat the cell. And the proteins from within the cell that, each type of cell kind of has its own protein that it sticks on that little pedestal.
Do the immunoglobulins help the cell know to put the bacteria pieces on it?
No, that is a function of the cell itself. Anytime antibodies interact with our cells are not in good shape. Something's wrong with them. A normal, healthy cell should not be attacked, should not have any interaction whatsoever, you know.
So up until that point, it's nothing.
Okay. Your civilians don't have anything to do with your army.
Yeah.
Okay. They don't, they shouldn't interact at all. But I found that one, and probably next year I will use parts of it and put it into my own. But I think I'll keep posting that one because he does make a good, he has a slight accent, I don't know what it is.
He emphasizes words in a weird way.
Yeah, that may be just that, but he has a little bit of a, I think it may be Massachusetts. Just that little, the way he says, it's not exactly Boston, but his O's and his A's are off.
Sounds just different.
Just a hair. But he's articulate. It was in the immune system. No, it was in the antigen body. Okay. So our MHC, just like he said, those are on, and MHC is, like he said, is on anything. So in humans, that's one thing that always confused me, is why it had two names. Humans, it's the HLA. So when you watch this lecture, you can substitute HLA antigens for, or HLA complex for the MHC. The probability that any two individuals have the exact HLA antigens is very low. That's why we use this. In fact, there are classes of these major histocompatibility proteins that line our cells on the exterior of our cells. You have HLA-A antigens, HLA-B antigens, and they're very, very confusingly named HLA-C antigens. And there are anywhere from 12 to 40 of each of those classes. So when they're doing, when you hear about them doing a paternity case, they don't actually use paternity case anymore, because what do they use instead? They use DNA now, because it's gotten down to where it's cheaper, easier, and it's much more accurate. But before the onset of DNA, we used our HLA complexes. Well, when they're going for a transplant organ to see whether it's going to be compatible or not, you know, all the TV shows, well, I'm the same blood type. Actually, blood type is not really important in a lot of your transplants. When's the only time you'll change blood types is if you've had a bone marrow transplant from somebody who's a different blood type. And I've seen it a couple of times. And you want to talk about freak out blood banker. Okay. So it's more important that these HLA antibodies match than our blood type matches. Why? Because this is what our recipient, what his or her immune system is going to be looking for. Okay, as he pointed out in the video, if it's holding up the right flag, that little I am me flag, okay, the little antigen it posts in there, that's the little I am me flag I was talking about. Okay, and if it recognizes that little flag, it's going to leave it alone. So the more similar to your particular antigenic profile it is, the better your body is going to accept it. Okay? A white person moving into an entirely Arabic neighborhood is going to stand out like a sore thumb. And if you've got a neighborhood that's not particularly friendly, that's going to be at risk, right? But if I've got somebody who is, you know, maybe Indian descent, they look close enough that they could blend in and not be noticed. Okay? It's kind of what we're looking at when we do organ transplants. We're trying to blend in with neighbors. We don't want anyone to notice us. So these HLA class molecules are the ones that we're going to be there, that they look at to see if we can make a transplant or not. The class 1 and 2 molecules bind to the foreign antigens so they can be recognized by the T cells. Okay? Remember I told you when that debris gets spit out at the end? That's kind of what part of that is. When it's ejecting that debris, part of that debris goes into those little notches on the MHC2 molecules so that the T helper cells and the other cells can recognize it and form antibodies against it. We told you that the ends of the antibodies, when they form, they're kind of mutable, right? They can wiggle. Well, it'd be much easier to try and make it the same exact shape if that shape ain't moving around. Does that make sense? And this slide right here is basically what that whole video was. Okay, that the molecules bind peptides within cells and transport them to be recognized. It puts them on the outside so you can see them. Class 1 molecules, the thing that you need to remember between class 1 and class 2, class 1 is presenting antigens that are from within the cell. Okay, it takes those infected antigens and takes them out of the cell and puts them. The class 2 molecules present antigens from that leftover digestive debris. Yes?
Between two people,
Mhm.
their HLA complex. You know how it said like alpha 1, alpha 2, beta 1, and beta 2?
Mm-hmm.
Are each person's molecules different, or like if you match cells, like for a liver transplant or something,
Mhm.
if somebody else had alpha 1 and 2 and beta 1 and 2 on their same, would it match, or are the actual molecules particular to you?
I don't know the answer to that question.
Okay.
I really do not. It's less about the actual holder, the little cup that attaches, and more about what's going into that little,
Okay.
what antigen is placed inside of that little holder, okay? But I do not actually know the answer to that question. I will see if I can find it. I'm not sure. I'll figure it out, okay? Like I said, the most important part though is what's being placed in that little, it's like that's your cup holder. What are you putting in the cup holder is the part that's going to be matching up.
Is that particular to yourself, or
That is particular to
very
yourself.
particular to yourself?
Yeah, that's particular to yourself. Those antigens that the cell pulls from the inside and sets in the little pedestal it's got there, those are the HLA antigens. Those are the A, B, and C classes that we're going to be using to make our match between donor and recipient. Does that make sense? Okay. Yeah, but the molecules themselves, I would guess, just based on what I know about biochemistry, it would be sort of like your PTH and his PTH. I don't know if they're unique to you or if there may be some variability between two. I don't think it's something we really put a lot of emphasis on.
Okay.
And that's a guess on there. MHC class 1 are on all nucleated cells. So what cells would not have these?
Red blood cells.
Red blood cells don't have MHC. So when we're cross-matching red blood cells, we're not looking at our HLA antigens because red blood cells don't have them. So when we're cross-matching our red blood cells, which you will do in blood bank, we're going to be looking at its own unique set of antigen presenters. Okay, and I'll let Barb get it. Basically what these do is they take endogenous antigens from within the cell, antigens from viruses. A mutated cell is just like a virally infected cell, is going to have antigens that are not normal to the cell. that's what's going to be moved out of the cell and placed for, basically to send out as a recognizer for our immune system. So a tumor cell is also going to put out abnormal antigens, depending on exactly what is wrong with it. But that's how our body recognizes tumor cells. You have tumor cells growing in you every day. But if you've only got one, pretty much the immune system just goes yuck and gets rid of it. What happens with cancer is they grow faster than the immune system can get rid of them. Or, in some cases, they find a way to mask that they are tumor cells. You know, those antigens, maybe the antigens aren't different enough, or the antigens don't get presented properly to the immune system. Yes.
Does that mean immunocompromised people have a greater risk?
Yes. Yes.
All cancers then?
Not all cancers, but many of them. AIDS was found because of Kaposi sarcoma, which is a cancer. So, and different immunocompromised people are, depends on what is causing immunocompromise, and we will talk about that when we talk about the various disease states, okay? Yeah. No,
The antigen is a piece of a protein.
yes, an antigen's a piece of a protein.
Is the one that's on that the cell sends out, is it the same one that's gonna coat, or is it different pieces?
It depends, sometimes it's different pieces, sometimes it's totally different proteins. And how it decides what goes up there,
Yeah.
I honestly have no idea. That, I'm pretty sure that if I keep watching that lecture series, that'll come up a little bit later, but I ain't got that far yet. Yeah, he does one on 2,3-DPG too, but it was way more than what you guys needed, so I took the 2 or 3 pieces that you needed. Not all cells have class 2 molecules. Okay. So class 1 molecules are kind of the civil guard. They're kind of the homebound monitoring systems. Right? These class 2 molecules are specifically on our macrophages, our B lymphocytes, and our monocytes. Which, as we know, the monocytes and the macrophages are basically the same cell. Okay. They're also on the dendritic cells, which we really don't talk too much about. They transport exogenous antigens, in other words, the pieces that are taken from what they ingest. Okay, so the bacteria that's floating around, they go gobble it up, yum, 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 little Pac-Mans, and then they spit out that extra. And when they spit it out, part of what they do with it is they take little pieces off the outside, they kind of like give treats to their dogs. Here, have a little piece of this. Okay, give them a taste for it, so to speak. So it takes and it destroys these exogenous, these things that are floating around outside of the cells. And that's the one big thing that you have to remember. Class 1 is dealing with stuff that is inside the cell. Okay. Viruses are rarely just free floating. They have to be inside another cell to be functional. Okay. So the virally infected cells, the tumor cells, those are basically going to be recognized because of the MHC class 1s. The things that come from, do you have a question? Okay. The things that come from outside, the bacteria, the things that can float around without support from the cells, those things are going to be presented because the antigens are going to be extracted from them when they're being eaten. And they're going to take little pieces of those antigens and put them on these external cells. And our circulating, especially our T helper cells, those circulating cells know to look on those little places for antigens. And if there's nothing there, then they just go on by. Well, this guy doesn't have anything for me today, so I'll just keep going. But if there's something there, then they're going to attach. They're going to clone off. They're going to force the other cells to clone, either into cytotoxic cells or into plasma cells. And it's always going to create some of those memory cells. okay? When we talked about the antibody comes down to a low titer, okay? It's not actual antibody that's floating around that gives you later immunity. It's technically a memory B cell. So it's not just like the antibodies just floating around in there. You have a memory B cell that knows how to make that particular antibody. Does that make sense?
Does it have to clone itself? Is it, I mean, sure it has a life cycle, so it, okay.
Yes, it does have to clone itself. Okay. Bacteria are the main type of antigen produced by, or that are presented by the class 2 molecules, which is why our fungus, our parasites, and our viral infections to a point, but our fungus and our parasites especially, those are the ones that, like our HIV patients, are particularly prone to. Most people don't have problems with fungal infections. I mean, dermatophytes, yeah, your skin. But as far as internal, as far as inside in the lungs or in any of your viscera, fungus doesn't really usually create too much of a problem for us. Parasites are not real common around here. Now you go to tropical areas and things are going to be a little bit different. So you need to remember which ones are the endogenous antigens, the things that you find within a cell, and the ones that are exogenous antigens, things that are found outside of the cell. And we already talked about that, right? Yes, we did. Smile and nod. Okay. So the next thing we want to do, we're going to draw pictures. I know you're all excited. So you're going to need a piece of paper and a pencil. We're going to do something active today to keep you awake and alive and alert.
Are we turning these in?
What?
Are we turning them into you, or is it just for us?
I wasn't planning on collecting them.
No, that's fine, because I'm just going to write on the back of my notes, so.
Oh yeah, put them on the back of your notes. Yeah. No, I'm not going to have you turn them. Okay. We're going to start with IgG. All you have to be able to do is draw rectangles. They don't even have to be very good rectangles. Okay, this is IgG.
The whole thing, or?
The whole thing. I gotta get. I like to draw wibbly wobblies on the end one, that way I remember which one it is. Okay. These chains here are our light chains. And they're smaller, so they're lighter, right? Makes sense? So this is going to be our kappa and lambda chains. These pieces right here are disulfide bonds. This is known as the hinge region because those little Y's can get farther apart and closer together. So this is the hinge region. And this is the constant region, also known as the FC. And this whole thing here is the antigen binding region. Do you need to know all of those regions? Ha-ha. If I put a blank one of these on the test and say label the regions, you have to be able to do that.
What is it?
This is it's hinge. I'm sorry, yeah, that's really bad writing. Is that better?
What is in the top and bottom boxes?
Light.
Okay.
Those are the light chains. So these are the heavy chains. So the smaller ones are the light chains, and these are the heavy chains.
Does say
I'm sorry, what?
the little line between the light chain and the heavy chain, what is that called?
Those are disulfide bonds.
Right.
Okay, this right here. We'll make our disulfide bonds orange. Okay? The orange ones are disulfide bonds.
And the bottom is just the constant.
The bottom area is the FC area. That in our bodies can be attached to a cell. And sometimes these antibodies are carried around on the outside of cells. When we're doing our testing, we can attach it to other things.
And what was that? The whole thing, what is it?
This one's IgG.
Under constant, what does the F0 stand for?
FC,
Oh.
it stands for the constant region.
What's FAB?
The antigen binding region.
So the FAB is the, okay, yeah.
Now that is going to be the same on, the regions are the same regardless of which antibody we're looking at. Okay, so like when we're talking about IgD, the antigen binding sites and the constant site, those are going to be the same. It's still all the same heavy chains, the light chains, and the disulfide bonds. The structure of each one is just a little bit different. And actually, on IgG, this is kind of two, I'm going home. This is kind of two proteins put together. Okay. So this is our IgG. These are also sometimes known as the variable region. All right, so we've got that all labeled up, yes? All right. So really bad at art. This is not a disulfide bond. It's just an area. Okay. Okay. There's our IgG. Now, if we're going to make, it has basically what we call 3 constant regions, okay? These are just regions on the protein, okay? This is C3, constant 3. This is constant 2. And this section here is constant section 1. So it has 3 constant sections and then a variable section on it. If we want to turn this into IgE, what we do is we add a fourth constant section, making this IgE. So the only difference is this is IgG, this is IgE. We add an extra constant section.
So is that another protein then? Added?
It's just another section of protein.
Alright.
So
Another section.
IgG and then IG.
Here's IgG.
Then IgG.
Add another constant region onto, or add another constant protein onto the constant region. Add another section of protein onto our constant region, and we have IgE. Take off. To make it IgD, this disulfide bond, instead of being straight across, is diagonal. And what does IgD do?
You don't know.
Well, maybe it might sort of. It has to have function. The body does not waste energy making something that does not have function. It just, I don't have a definition for the function yet. And I found a couple of different articles that tell me exactly what it does. And they're contradicted by a couple of other articles that say the same thing. So I haven't found a good definitive thing. What does IgE do?
Secretions.
What does IgE do? Allergies. Okay. So for IgD, I add this extra, I change this disulfide bond and I make it diagonal. As you can tell, I'm really good at drawing. IgA, does anybody remember what I told you? Does anyone remember what I told you IgA does once it gets out into the secretions? It hooks up as a dimer. Okay? So IgA, what you wind up with on IgA, you get one of these days I'll need to draw there, sorry guys. And this linkage right here, get kind of a little spiral linkage using the J chain that hooks this into a dimer formation. Okay, you're gonna have the same exact things here. We're gonna have C3, C2, C1, and then the variable region. So this is gonna be IgA.
It also has diagonal. Disulfide.
Yes, it also has diagonal disulfide bonds. This is sort of- No, it doesn't. I don't like these pictures. No, the one with the diagonals is D. Okay. So but the thing with IgA is it's going to be kind of wrapped with this protein to hold it together. So we've done G, E, A, D. I'm not drawing M, we will be here until Tuesday. So if I take this same kind of linkage and add 3 more to it, so that I have a pentamer, so that I have 5 of these IgG molecules, 5 of our basic monomers put together, hooked up with a J chain. That is going to be my IgM. Okay. Now, like I said, if I here. There we go. There's the other monomers. If I put 5 of them together, I wind up with my protein.
So on that end, what is those V?
Those are the variable regions, that's the FAB. This is the little wibbly wobbly region that adjusts and changes itself. What happens is when this is presented to a protein, everybody got this?
No.
Okay, I will wait.
So those don't hook up like that side, like it don't have disulfide, little bonds on that side?
Yeah, it does, I just didn't draw them, I'm sorry. Yeah, each monomer is identical. Yeah, there's our IgM.
And this is the IG.
That's the IgA. The dimer is the IgA. To make the IgM, you have to hook 5 of them together.
Are
Yes.
you using 5 different individual J chains or one J chain? and
One big
one-
J chain binding it all together at the bottom.
Too much.
It's a really good representation.
There's M. Now you see why I didn't draw it.
It has diagonal.
Pardon?
It has diagonal.
I know it keeps showing these as,
It does.
Does that have a C4?
Right, that's what it looks like, that C4 too.
it does look like it has a C4.
What's the A look like? In that, do you have an A on, like? the
Yeah, I
same-
got it, hang on, I'm looking.
Oh.
Here's the A.
Explosion.
And that one just has a C3.
It does have, looks like there is an extra on this, on the IgM, it does have a C4. IgE though has, okay, the only reason I stress those things is when it comes to the test, that's what you're gonna be looking for to be able to differentiate them. The IgE is gonna be a monomer with a 4th constant region, okay? I won't put IgD on there, how about we just go with that, okay? And I will look into the diagonal disulfide bonds. I'll talk to Jean about it, because this is her
It looks
notes.
like A only has 3.
So you
Probably
wanna
just
put
'cause
which
that-
one on there?
Little chain.
Wait, so
Probably just because Diane's a really, really,
No, in that.
oh, in here.
Yeah, look.
Yeah, it does only have 3 constant areas. These are IG.
No, 3 bonds.
3 bonds.
Right behind your hand,
Down, there's no bond
yeah,
Yeah.
back towards you.
Here?
there. Yeah.
There is a bond here, it's just hidden behind the thing. There has to be a bond between the 2 sides, or the thing would fall apart. There is a disulfide bond that goes here. Now, it may be linked with the J chain.
So you gonna take which one off on the test?
I won't ask D.
D? Okay.
I won't ask D. Because you're right, all of these pictures look like they're diagonal disulfide bonds.
Well, does.
But see, here's
G
the,
look diagonal?
here's the regions on here. It could be what Jean copied to make these too, so I don't 100% know. I never really paid that much attention to it. I just remembered E has an extra piece. D has a diagonal disulfide bonds. A hooks up as a dimer. and M is a pentamer.
So this is the only drawing we would have to label. You wouldn't.
This
make
would
us-
be the only drawing I would have you label.
Okay.
Yeah, you wouldn't have to label the regions on the other one, I might, but if I draw one that has, if I draw a monomer that has an extra, because truthfully, this should have, these are the variable regions, this should have C constant place 1, constant region 2, and constant region 3 are not labeled on this particular drawing, but I happen to know that's the one that's on the test. So when you go, and this is not a structure, this is the hinge region right here. So this circle is not a structure. That circle is just, we circled the thing.
Will we have to know gamma and alpha like that? Yeah.
Oh, if you keep looking, that's the rest of it. Gamma, alpha, mu, epsilon, delta. That's the 5 kinds, G, A, M, E, and D. And yes, you will have to know that there are 5 kinds. But
Can we get a copy of that?
yes. How many? Are you guys here tomorrow? Okay, I will Xerox it tonight. And you? Yes.
So the double lines in the hinge region, that's another disulfide bond.
This is a this is a disulfide bond, these are disulfide bonds, and this is a disulfide bond.
So on the light chains, are they both going to have a kappa and a lambda, or does it depend on which IG molecule it is?
I'm not gonna answer that question because I think I know,
Okay.
and I don't wanna answer it wrong.
So one of them, it has at least one of them.
I think it has both,
Okay.
but let me look and double check that before I say something, okay? But these kappa and it may be based on which one, so I will look up that information. Hey, I am not afraid to tell you I don't know stuff, so when I don't know, I will find the answer and get it to you. This is exactly how I was my first year of hematology. Guess what? If I send an announcement with that information, will you want it? Do you check your student emails on any kind of regular basis? Yep. I'll do that. I'll pop an announcement. If I don't get it tonight, I will pop it out to you tomorrow.
Is our first test gonna be with you on Monday?
What does it say on your schedule?
I throw them out because they never are accurate.
Did we get a schedule
Hang out here for a
for
minute.
serology?
Hang out here for a minute.
It's attached to the,
Kim.
For today and yesterday.
I
Okay.
got the PowerPoint report.
Bacterial antigens, I'll just redo it later.
Okay.
Okay.
Just immune and antibodies.
Just the immune system and the antibody complexes. By Saturday, when I head to work at 1, and I won't guarantee before that, but by Saturday at 1, for a change, I will send out a very pointed study guide. Okay? Very pointed, i.e. study the crud that's on the study guide, because that's all I'll put on the test. All right, but you'll have to give me till Saturday to get that out. As a overall general one, the immune system, make sure that you know the difference between passive and active immunity. Just so this will give you some review. But I don't want to go on to another, I actually don't want to go on to the next, I don't want to go on to another lecture, so let's do a quick review for the test now. We'll slip through the stuff and I will point it out.
Yours was mostly just multiple choice, right? Or did you have short answer on your tests?
What's that?
Yours is just multiple choice, or did you have short answer too, like definitions?
I added short answer because that was something I needed. So, and what I'm going to do is I'm going to record my own because that way I know exactly what I said. Yes.
Are we going to need to review our normal values we did for you? For coag and
Oh, for heme?
heme.
No, I don't really see that as going to be
too big of an issue?
No. I mean, before you take the final exam, of course you're going to, but.
Well, yeah.
I'm not gonna, I'm gonna record it for 10%. Okay. All right, so for the immune system, make sure you know the terms that are on there. Antigen and antibody especially, I would say that vocabulary would definitively be something that would be open to short answer. Make sure you know the difference between natural and innate immunity and acquired or cell-mediated immunity, and which one is active and which one is passive. We're on the immune system, I apologize. Yeah, I'm just gonna slip through these slides real quick and kind of point out the
Slide
big
4
pieces.
on immune system.
So immunology, make sure you know the terms. Make sure that you know that natural or innate immunity is passive and not specific. It's very general. And that acquired or cell-mediated is active and it is specific. Okay, so it's against a particular thing. Opsonization, just remember that that's basically coating the foreign substance so that the immune system recognizes it better.
Is that the first time the immunoglobulins will have contact with the cell?
Pretty much. There's a list of all of the things that are our external defense system. Again, that would be something probably, which of the following is not part of the passive immune system? Or part of the external? Know the steps of the inflammatory reaction and its significance to health. That it is basically, it starts with redness, swelling, heat, and pain. It should go away shortly, but if it persists, it can be a problem later on. Know what an acute phase protein is. And know particularly with CRP that it responds within 4 to 6 hours and goes away within 48 after the infection's cleared up. Know what phagocytosis is, that's pretty simple, straightforward. Complement, we'll talk a lot more about it later. So if I mention it on this test, it would simply be as complement is part of the inflammatory reaction, something along those lines. Okay, I'm not gonna ask a lot of questions about it right now. We will talk about it quite a bit more later. This should be reviewed, the lymphoid organs. Know where T cells mature, where do B cells mature. CD markers, I will only hold you to the ones on this slide. 2, 3, 4, and 8 are T cells. 19, 20, 21, and 40 are B cells. We all know that there are others, don't we? Smile and nod. But for this test, I will only hold you to these. The most important are the CD4, CD8, CD20, and 40. Again, this should be review how we identify those CD markers. Pretty much just know that we use flow cytometry to ID the CD markers. I'll get more into the fluorescence and the how-tos when we're talking about using antibodies as their as a test methodology. Know the difference between a monoclonal and a polyclonal antibody. Know the difference between a monoclonal and a polyclonal antibody. And what is the difference?
One will make a single antigen versus polyclonal.
Could be different ones. They're made by different B cells. Monoclonals are made from a clone. Okay, so you basically have identical plasma cells making those antibodies. They are made from a single clonal population. Polyclonal antibodies are made by a variety of lymphocytes, not necessarily clone brothers. Know that forgot we had one. Know that antibody producing cells are plasma cells, and that should be again reviewed from hematology. Know that between 15 and 35% are B cells, so about a quarter are B cells, about 3/4 are T cells, and there's a little sliver that are NK cells. T cells, again, know that there are helper and cytotoxic cells, that the T cells are split down into those. NK cells, basically know they exist and they're not T cells, they're not B cells, and as they're important in particular disease states, we'll talk about it, they're found in spleen and peripheral blood. They kind of float around looking for the bad guys. And then I will probably focus most strongly on this for antigen antibody. This is where I'm really going to pull probably the bulk from, because this is kind of the meat and potatoes of immunology. So you're going to, well, first of all, again, you're going to need to know antigens. So what is an antigen and what is not an antigen? What is an antigen? A protein or a piece of a protein. List some things that are not antigens. And well, antibodies can't actually be antigens, believe it or not. We'll get you really confused with that later. Right, organs, full organisms, full organs, full cells are not antigens. It's protein pieces. What is the most common type of chemical that is, proteins are our most common antigens. What's the other one that's next? Carbohydrates. We can have others, but it's usually proteins. Just because proteins are fairly regimented, they're structurally regimented, they are chemically regimented. So it's kind of easy for our little wibbly wobbly end. Because if you think about, here's an enlargement of just the variable end of our antibody, because y'all know how good I am at drawing, right? So what happens is we have a protein, and we all know that proteins are linked strings of amino acids, which then kind of curl up on themselves. So they have a primary, a secondary, and a tertiary structure. The primary structure is what amino acids are in them. The secondary is what order those amino acids in. And the tertiary is how they all kind of wobble up into like a little globule ball. Okay? So what happens is my particular piece of the protein that is serving as an antigen, let's say it looks like that. Okay? What's going to happen is this is going to scoot over there, and it's going to go over here, and its end is going to change shape. And it's going to look like that. Now this is obviously going to be much more convoluted, okay? But it's going to go so that it's exactly that shape. So what then happens is this antibody with the variable N goes out looking for anything that's that exact shape and hooks onto it. Okay? So that's why we call this the variable N, because it shapes itself into it. Okay? It's like a little blob. Right? Know what an epitope is? And an epitope is the particular section of an antigen that is recognized by the immune system. There is one epitope for each antibody, and each antibody has its own epitope. Be able to define the types of antigens. A hapten, remember I told you, that is not an antigen in and of itself, it's too small, but a lot of times it'll combine with something else. Okay, so it might be, I don't know, an aluminum salt piece that is attached to a globule of some sort. I don't know, making things up there. But basically, it's a piece of something that by itself isn't big enough to be a stimulant, to be an antigen, but it hooks up with something else combined with a carrier molecule of some sort. Autoantigens are something against yourself. Alloantigens are from another member of the host species, so in our case, other humans. And heteroantigens are from another species. An adjuvant is not necessarily something we find in nature, it's used in vaccines, and it's something that increases the attraction to the immune system. Okay, it helps to boost the immune response. We talked about MHC today, and I kind of stressed as we were going through, you need to know the difference between class 1 and class 2. You need to know that they are found on nucleated cells and nucleated cells only. That with the class 1 molecules, it's things that are inside the cell that are presented. The class 2 are found only on limited cells, the B lymphocytes, monocytes, macrophages, and that these present external antigens or exogenous antigens to the immune system. So pieces of bacteria, pieces of what these cells had for lunch. Okay. I guess that means class 2 deals with vomit. That's nice. Okay. Anyway, be able to define an antibody, know the 5 classes of antibodies, and what are the 5 classes?
Gamed.
I think I've heard all of them, G, A, M, E, and D. Okay? Make sure that if I just ask you to list them, you can list them.
You know the letter or the names?
Gamma,
The real names?
beta.
I don't think I ever hear them referred to as anything other than IgG, IgA. They're not referred to that way. So let's just stick with IgG, IgA, IgM, IgE, IgD, and GA, ME, and D. Yeah. They're Greek letters. If you had to go through and think about them, you could. But I don't, other than gamma globulin, know that that is IgG. Because that one, we do see that written in text and in literature. And it is referred to, especially by some of the older doctors and nurses, as gamma globulin. So that's the only one I'll ask you to know by name. The other ones, we just really don't refer to them by name. Be able to label the structure of an antibody. It will tell me that they are connected by disulfide bonds, the light chains, the heavy chains, the constant region, the variable region, the hinge region. Okay, you need to be able to label that diagram. You're going to need to, if I give you a picture, be able to tell me if it's IgG, IgA, IgM, IgE, and I won't put D until I get that clarified. Okay. So GA, ME, and D. The IgG structure of that one, it is a simple monomer. Then if you take the IgA, it becomes a dimer linked with a J chain. And where do we find IgA? In secretions. IgA is the one that's associated with secretions. So when I'm asking for function, that's the function of IgA. And in the secretions, it hooks up as a dimer. IgM is what shape?
Pentamer.
It's a pentamer. There's 5 of them hooked together. That's why it's also known as a macroglobulin, because it's big. It's macro, it's really big. Okay. IgE has an extra constant chain on the end of the monomer. Those are kind of going to be your clues.
The J chains on there, is those little lines?
The J chains are kind of they're like a protein chain. It's kind of like a piece of yarn that wraps
Okay.
around it to hold it
Yeah,
together.
that little-
Yeah,
okay.
just kind of wraps around it to hold it together. Know that there are subclasses, and there are only 4, I looked it up. There are 4 subclasses of IgG. There are 5 tests in the IG subclass panel because it gives you a total too. That's where I got confused. So you don't think I'm a total idiot. Know the function of each one, we already talked about that. Know which one crosses the placenta and which one does not.
IgG is the only one that does, right?
IgG is the one that does. I think that sounds like enough for a test, doesn't it? So that's kind of what will be on the test. And as far as I know, it's going to be Monday. That's because she told me when Barb was done with her test, we were just going to put the serology tests instead. Okay, I've got the recorder. Everybody got the recorders turned off now. I gave you the bacterial antigens lecture. I have it already on another test. I really don't want to have to rewrite the test to add it. So what I'm going to do is we will do a brief review like I just did with these. We'll do a brief review of it when it's time for it to come up on the test. Okay, it won't hurt you to go over it again. But I think there's enough to go on with just these 2. Let's see, I don't know when my next time my lecture is, but I know pretty much starting next week. Next week is week 6. There's a lot of lecture coming
I
up.
would say, I think she said you're doing a lot of lecture next week.
I am, next week and the next couple of weeks. So